Good morning, student. I am Mr. Vinod Thombre. We are going to study the subject plant physiology, and in that, the important topic from plant physiology is photosynthesis. Now, first of all, what is photosynthesis? If we split the word photosynthesis into two different words, which is photo and synthesis, then we easily understood the meaning of photosynthesis. Photo, that means light, synthesis means production, hence synthesis of food with the help of light in plant is nothing but photosynthesis. And all the biochemical reactions that come into the category of synthesis are called anabolic reactions. Now see the definition of photosynthesis. Conversion of light energy into chemical energy is called as photosynthesis. This is the most simplest definition of photosynthesis. Now the photosynthesis is generally takes place in the chloroplast. Now to understood how the photosynthesis takes place and what kind of proteins they are involved in the photosynthesis, for that we must know about the structure of chloroplast. If we study the ultrastructure of chloroplast, then we know that the chloroplast is a bilayer membrane cell organelle which is generally found in plants. Chloroplast, which has a intermembranous space because it is a bilayer membrane cell organelle, the outer envelope and the inner membrane and the area between outer and inner membrane is called intermembranous space. Then in the intermembranous space, the surface which is accumulated is called stroma, and in that the thylakoids that are present and thylakoids that link by a structure which is called stromal lamellae, which is the site of photosystem 1, and the stack of thylakoid is called the granule lamellae, and which is generally the site of photosystem 2. Then if we study the single structure of granum, which is called the stack of thylakoid, then we able to know the structure of thylakoid, that it has thylakoid lumen and the outer portion which is called stromal lamellae. Now first of all, we study the composition of photosystem 1 and photosystem 2, which are involved in photosynthesis. Photosystem 1 and photosystem 2 composition. The discovery of ray drop and the immersion enhancement effect has led scientists to suggest that photosynthesis is driven by two photochemical processes. These processes are associated with two groups of photosynthetic pigment called as pigment system 1 and pigment system 2. Wavelength of light shorter than 680 nanometer affect both the pigment system, but wavelength longer than 680 nanometer affects only pigment system 1. We called it as a photosystem 1. Now the composition of photosystem 1, or which is also called as pigment system 1. Butler in 1966 found that the pigment chlorophyll A exists in two forms, one having absorption maximum at 673 nanometer, which is called chlorophyll A673, and the other at 683 nanometer, which is called chlorophyll A683. Clayton in 1966 discovered another form of chlorophyll A which has absorption maximum at 700 nanometer, and this chlorophyll is called P700, or the photosystem which absorbs the 700 nanometer life, which is utilized for the photosynthesis. Goenji and Rabinovich in 1960, Mears and French in 1960, Jones and Mears in 1964 have shown that light energy for pigment 1 is collected by chlorophyll A 683, that means the chlorophyll A which absorbs the 683 nanometer, chlorophyll A which absorbs the light of 695 nanometer, P700, and the carotenoids also contain chlorophyll B. Pigment system 1, that is photosystem 1, consists of a central core complex 1, that is CC1, and light harvesting complex 1, which is called in short LHC1. That is the composition of photosystem 1. See the structure of photosystem 1. Structural model of the photosystem 1 reaction center. The structure shows component of the photosystem 1 reaction center, which is generally organized around two major proteins, photosystem PSAA and PSAAP. Minor proteins PSAC to PSAN are labeled to C to N. Electrons are transferred from plastocyanin to P700 and then to a chlorophyll molecule, A0 to phylloquinone, A1 to the FESX, FESA, and FESB, FES center, and finally to the soluble iron sulfur protein. Ferredoxin FD, the last electron carrier molecule which reduces the NADP to NADPH. The B diagram is the side view of one monomer of photosystem 1 from the cyanobacterium Cyanococcus elongatus at 2.5 angstrom resolution. The stromal side of the membrane is at the top and the luminal side is at the bottom of the figure. Transmembrane alpha helix of PSAA and PSAB are shown as a blue and red cylinder respectively. This is the structure of photosystem 1. The core complex consists of their respective reaction center associated with proteins and also electron donor and acceptor. The light harvesting complexes contain antenna pigment associated with proteins, and their main function is to harvest light energy and transfer it to their respective reaction center. The location of photosystem 1 is generally present in stromal lamellae. Then the second photosystem, which is called pigment system second or photosystem second, contains chlorophyll B and some form of chlorophyll A, such as chlorophyll A, which absorbs 662 nanometer light, chlorophyll A670, chlorophyll A677, and carotenoids. A very small amount of special form of chlorophyll A, which is called P680, that is the pigment system absorbs 680 nanometers light, so it is called P680, constitute the reaction center of pigment system second. In case of the red and blue-green algae, the pigment system contains phycobilins in place of chlorophyll B. Pigment system second consists of a central core complex second, like photosystem 1, which contains central core complex 1. Photosystem second contains core complex 2 and the light harvesting complex 2. The core complexes consist of their respective reaction centers associated with proteins and also electron donors and acceptors. The light harvesting complexes contain antenna pigment associated with proteins, and their main function is to harvest light energy and transfer it to their respective reaction centers. The location of photosystem second is present on granule lamellae. See, this is the structure of the photosystem second reaction center from the cyanobacterium Cyanococcus elongatus resolve at 3.8 angstrom. The structure includes the D1 and D2 core reaction center protein, the CP43 and CP47 the antenna protein, cytochromes B559 and cytochrome 550 the extrinsic 33 kilodalton oxygen evolving proteins, PSBO and the pigment and other cofactor. A diagram. And in B diagram, view from the side view parallel to the membrane plane. Then the functioning of photosystem 1 and 2. Photosystem 1 produces a strong reductant P700 and weak oxidant. It can therefore reduce NADP but cannot photolyze the water. It reduces NADP to NADPH. Photosystem second produces a weak reductant, that is P680, but a very strong oxidant, because of which it can cause photolysis of water. It causes the oxidation of water, which is generally the complex which is called oxygen evolving complex, which is present just below the photosystem second, and which causes the oxidation of water to split water into the 4H plus ion and one oxygen molecule by breakdown the two water molecule. Thank you.